right, continuing here in chapter 2, learning a little bit more about some of the terms we mentioned in the first one and more about pH. So first of all, inorganic and organic compounds. So we already kind of mentioned this whenever we talked about the cofactors, how some are inorganic and some are organic. And if they're organic, they're called coenzymes, like our vitamins. So what does that really mean? So inorganic chemistry are things that do not have carbon-hydrogen bonds. Okay, so water is inorganic, it's hydrogen and oxygen. Carbon dioxide, CO2, has carbon, but no hydrogen. So again, inorganic. All of our ions and metals like iron and zinc, again, inorganic. Organic is where we do have carbon, hydrogen substances. So again, carbon monoxide, carbon dioxide, not examples because they don't have the carbon-hydrogen bonds. But as long as they do, we refer to them as organic molecules. So talking here a little bit about some of our inorganic. So water, extremely important to our cells, of course. We already mentioned in our last recording, it is polar because it has polar covalent bonds. So it has a slight positive end where that hydrogen is and a slight negative where the oxygen is because the electrons are shared, but not shared equally. All right, so it's polar. Then the term's hydrophilic, which just means water loving. And then we have hydrophobic, which is water fearing. So things that like water, like salt, hydrophilic. Things that don't, like oils, hydrophobic. A couple other terms here related to water, cohesion and adhesion. Cohesion is the attraction of water to water. So one water molecule to another water molecule. So surface tension is a good example of this. So like beads hanging on a spider web where the water is forming bubbles and beads because it likes each other. They were attracted together by those hydrogen bonds. And then adhesion is the attraction of water to another molecule besides water. So like water to a sugar, that would be adhesion. Okay, so some of the other properties, water has the ability to stabilize our body temperature. So of course, whenever we get hot, we sweat. The release of that water that evaporates removes that excess heat. So it helps stabilize our body temperature. It also provides some protection. So it can be a lubricant. So we think about like our joints, and that liquid then forms kind of a cushion around our brain. We have that cerebral spinal fluid that provides some cushion to protect from trauma. And as we talked about with dehydration and hydrolysis reactions, we need that water for those chemical reactions. Okay, we also talk about it whenever we're mixing things. So solutions is the mixture of a liquid, gases, or solids. So in that case, we have a solvent, which is what's dissolving a molecule. The molecule being dissolved is solute. So if we have salt water, for example, water is the solvent and it's dissolving the salt. So salt is the solute. And then the term osmolality refers to the number of particles dissolved in 1 kilogram of water. So the concentration of solutes in a solution. So we'll talk a lot about this again whenever we talk about osmosis in the next chapter. Now, water is also important to determining our pH and our acids and bases. So just first of all, an acid is a proton donor. So it is donating hydrogen ions. So again, ions have the positive or negative charge. Again, quick review of our terms from the other recording. This is a cation because it's positively charged because it lost an electron. Chlorine is an anion. It is negatively charged because it gained an electron. Okay, now in this reaction, hydrochloric acid, so again, it's a reaction. We have our arrow here. And so our reactants in this case, hydrochloric acid on the left, forming the products hydrogen ions and chlorine ions. All right, so hydrochloric acid is an acid because it is donating hydrogen ions. The opposite of that is a base, which is a proton acceptor, or anything that binds and accepts hydrogen ions. So for example, sodium hydroxide. So sodium Na, and then hydroxide is our OH. Okay, so again, it dissociates into sodium ions and hydroxide ions. Again, hydroxide is negatively charged, so it's an anion. Well, it is attracted to those hydrogen ions that are positively charged. So these hydroxide ions come down here, pick up some hydrogen, and that's going to form water. So that hydroxide of sodium hydroxide is accepting or absorbing, taking in some of the hydrogen ions, so it is a base. Now we have a pH scale that goes from 0 to 14. Now this is a negative log scale, and we are measuring the concentration of hydrogen ions in a solution. Now this, because it's a log scale, is a little bit backwards. Okay, so a pH of 1 is actually 10 to the 1 to the 10 to the 0, which is just 1. Okay, whereas a pH of 4 is 1 times 10 to the negative 4, which means we have 0.0001. So we move that decimal 4 places. But as you can see, this number is much smaller than 1. Okay, so even though a pH of 0, that number is lower than, of course, a pH of 4. A pH of 4 means we have less hydrogen ions than a pH of 0. And then the least amount of hydrogen is at a pH of 14, which is our highest pH value, but the lowest amount of hydrogen ions because it's 10 to the negative 14. So we move this decimal 14 places from 1. So it's an extremely small number, small amount of hydrogen ions. Now we also have a couple terms with this. So neutral is a pH of 7. So right in the middle between 0 and 14 is 7. So that is a neutral pH, meaning there are equal hydrogen ions and hydroxide, which is the OH. Now acidic would be a pH of less than 7, which means we have a greater amount of hydrogen ions. Again, pH of 1 compared to 7, we have more hydrogen ions than here at 7, because again, that's the number of zeros we have in front of our 1, so it's much smaller. And then alkaline or basic is a pH greater than 7, but that means now we have more hydroxide and less hydrogen ions. All right, now why is pH so important? So first of all, our normal pH, except for in the stomach, our normal pH in our blood, for example, is 7.35 to 7.45. Anything, and that's a pretty small range, anything below that, whenever our blood drops below 7.35, that is called acidosis because the pH is lowered. So we're more in an acidic condition, and the nervous system becomes depressed, and we actually become kind of disoriented and even comatose. So again, just a slight variation, 0.1, so from 7.35 to 7.45, anything above or below, and we're in trouble. So acidosis is below. If it goes above 7.45, we call that alkalosis. Again, in this case, our nervous system still gets altered, but in this case, we get overexcited, becomes very nervous, and can actually have a seizure or a convulsion. So things start slowing down when it drops below 7.35, speeding up above 7.45. So because that range is so small, we need to have a way of preventing changes to that pH. And that's where these buffers come into play. In addition, we need salts as well. So the terms here, salt is anything consisting of a cation besides hydrogen ions and an anion. So thinking about ionic bonds, so we have a cation and an anion. So again, salt, sodium chloride, but there are others as well besides sodium chloride. But again, it's just an ionic bond where we had a transfer of electrons and a cation is attracted to an anion. Now, a buffer is a solution that has an acid-base pair. And then in this case, the whole point is to resist changes in the pH. Okay, so when there's either acids or bases that get added, these buffers work in a reversible reaction to keep that pH pretty normal, to keep us from going below 735 or above 745. Now, the important buffers in our body, the bicarbonate, that's the one we talked about with the carbon dioxide and water that combine to form carbonic acid that then dissociates into hydrogen ions and bicarbonate. We also have phosphate is a buffer system, and some of our proteins that are made up of amino acids, they actually are important buffers as well. Okay, so what happens then with a buffer? So again, these are acid-base pairs. So in this situation, if we have an acid that gets added, so we eat a tomato or we drink coffee that's very acidic, it's going to start donating hydrogen ions. That's the definition of an acid. It increases the hydrogen ion concentration. Well, if it goes too much, we're going to drop below the 735. That's going to lead to that acidosis. We don't want any of that. So we need a buffer. And what that buffer is going to do is part of that buffer would bind, so the base would bind to those extra hydrogens in order to balance them out. So they're going to absorb or accept all of those extra hydrogen ions. So here with our buffer being added, again, those hydrogens combined to form, combined with our base to form what's called a conjugate base. So when the acid is added to that solution, the base sucks up and removes the hydrogen ions to take that pH back to normal. All right, now just a little bit about a couple other inorganic molecules. So oxygen and carbon dioxide, of course, talked a lot about water, but the other two, oxygen and carbon dioxide. Oxygen, of course, very important for breathing, but the reason for that, we need that oxygen because it's the final electron acceptor when we're getting energy. So aerobic respiration, whenever we're breaking down molecules to get energy, we need oxygen there to remove those electrons. And then CO2, carbon dioxide, this is a product of catabolism. So again, the breakdown of proteins and carbs to get energy. So it's a waste product that needs to be removed. That's why we exhale it from our lungs. But also, again, that bicarbonate buffer system, it combines with water and forms hydrogen ions that affects that acid-base balance. So that's why we can't hold our breath. So if you hold your breath, not only are you not letting oxygen in, but all that carbon dioxide is building up, forming carbonic acid that leads to that acidosis, and it causes our body to react by breathing to release that CO2. Right, now switching into our organic chemistry. Now this is again where we have those carbon-hydrogen bonds. So we have carbon atoms held by those covalent bonds. These are going to form the backbone of a lot of large biomolecules like carbohydrates, our nucleic acids, proteins, and our lipids. What joins them together is they all have these carbon backbones, and then functional groups get attached to make them unique. So if we look at the four major organic molecules of our living organisms, again, we have carbohydrates, we have lipids, we have proteins, and nucleic acids. So a little bit about each of these. Carbohydrates, as the name kind of implies, they have carbon and hydrogen. It gives us carbohydrate. But also oxygen in there. So they are made up of monosaccharides like glucose. If we have two or more monosaccharides, that forms disaccharides. Then if we have a lot of monosaccharides like starch or cellulose, that forms polysaccharides. Now, carbs are important for energy. That's usually what we think about with carbohydrates. But they're also really important for structure. So cellulose builds plant cell walls. So that's made up of glucose as well, so very important to providing structure and support. But also cellulose for us, we can't break it down, so it adds bulk to us. So high fiber diets add bulk to our digestive system to help elimination of waste. And these are water soluble molecules, as most of them are. Now, lipids are our exception. They are not soluble in water. They are insoluble. That's what unites them into this group. So it's very diverse. They have lots of different molecules. That what they have in common is they're hydrophobic. They don't like water. So these have also carbons, hydrogens, and oxygens. But again, they arrange themselves in non-polar bonds, so they don't like water. And there are quite a few functions of our lipids. They can help provide protection, so like a cushioning around our organs in case of trauma, just like our water and our fluids. They can help with insulation. So we think about our polar bears and things that hibernate. They store up lots of fat. It can regulate our physiological regulation. So some of our hormones are lipids. They're the main part of our cell membranes. We'll talk more about phospholipids to form our cells. And they also are used for that energy storage. Now, proteins, also carbon, hydrogen, and oxygen, but these guys also have nitrogen in them, sometimes some sulfur as well. And these have lots of different functions, probably the most diverse. Doesn't make them the most important, but proteins do a lot of different things. So they're important in regulation. They help with transport, so moving molecules in and out of cells, as well as just moving things through the body. They also help with protection as far as like antibodies and protecting from infection. Our muscles contract with the aid of proteins. Structure, they can be broken down to give us energy, and our enzymes are another one. These are proteins as well. And then finally, our nucleic acids, so carbon, hydrogen, oxygen, same as the others. Also nitrogen, like we'd seen with proteins, but then phosphorus is kind of unique to these. Now, these really only have one function, well, two. Genetic storage, so DNA and RNA that stores our genetic information, but then also that ATP that we talked about with energy, that is provided by these nucleic acids as well. Just a little bit more here specifically about the phospholipids because they are so important to making our cell membranes, and they'll be important in our next chapter talking about movement in and out of cells. So these are lipids, meaning they're mainly, again, hydrophobic. They don't like water, but they do have a slight hydrophilic part as well. So they have what we refer to as a hydrophilic head group and then two hydrophobic tail parts made up of those lipids, those carbon hydrogens who share electrons equally. So those are non-polar covalent bonds, so there's no attraction then to water. But they do have some oxygen and phosphorus as well as some nitrogen here in the head that do form bonds with water because they form non-polar, or they form polar covalent bonds. So they have this hydrophilic kind of head region and then these two hydrophobic tails. These are going to line up in a way to form our cell membranes. Now, a little bit more about our proteins. Again, these are made up of amino acids. They're the building blocks. So we'd shown this example. Again, this is that dehydration reaction because the hydroxyl group comes off of one amino acid, the hydrogen off of another, that forms water. But now this carbon and this nitrogen need to form a bond to fill their outer orbital, and that's where we get the formation of this peptide bond. Now, the structure of proteins is extremely important because it determines their function, and there can be up to 4 layers to their structure. So the kind of starting point for their structure is called the primary structure. That's the initial shape they take on, which is just the amino acid sequence. So there are 20 different amino acids. The order that they line up in and the number of total amino acids is different, and that forms what's called the primary structure. And ultimately, that's going to determine all the other structures. So then this chain of amino acids over here it's going to twist and bend using hydrogen bonding between those amino acids into different twists and bends, and that makes up our secondary structure. Then ultimately, each of those chains forms what's called the tertiary structure, or three-dimensional shape, and therefore function of the protein. Okay, now if a protein is only made up of one chain of amino acids, tertiary structure is the highest level of structure. However, some, like hemoglobin, is made up of 4 different chains of amino acids. So the way that they all come together forms what's called the quaternary structure. And each chain of amino acids is called a subunit. Now, because of that structure ultimately determines function, anything that alters the ultimate shape is called denaturation, and that is going to change the shape and therefore function of the protein. So if a protein is denatured, it has lost or changed its shape, and therefore it is no longer functional. Now, our last one that we'll talk about is the nucleic acid, a little bit more about it. So the DNA is our deoxyribonucleic acid that's storing all of our genetic information. Its structure, again, determines its function. It is a double helix, okay, which is shown here. So it has 2 strands of what are called nucleotides. They run in opposite directions. And each nucleotide is made up of 3 parts. We have a phosphate, we have a sugar, and we have what's called a base. And there are 4 bases. So we have A, C, T, and G. As you can see, there's slight variations. A's and G's are a little bigger than the C's and T's. But they also exhibit what's called base pairing. So these hydrogen bonds, shown as these little red dots, those hydrogen bonds are going to actually join these strands of nucleotides, and they always come together in a pattern or a pairing in which C always pairs to G, A always pairs to T, and we say that they are complementary. So it's not the same sequence on both sides, but they complement, they fit together. So we have these 2 strands of nucleotides. They run in opposite directions. They're forming hydrogen bonds between the bases. That pairing is always A and T, and then C pairs to G, making them complementary. And then ultimately they twist into this double helical structure. Now, the other nucleotide of importance is that ATP that is our energy currency of this cell. Again, it's a nucleotide, so it has a sugar, it has at least 1 phosphate, and then the base, which in this case is the A, the adenine. And then this is called triphosphate because it has 3 phosphates. And this is, again, that energy currency of our cells. And energy is formed and stored in the bonds that hold these phosphates together. So by breaking one of those bonds, it releases energy. If we need to form the bond, we need energy. So the reactions go like this. So in catabolism of glucose, so the breakdown of glucose, that releases energy. That energy is used to add a phosphate to ADP to form that adenosine triphosphate. So it's adding that 3rd phosphate on. Then if energy is needed, say for muscle contraction, ATP is then broken down. We release that 3rd phosphate. Whenever that bond is broken, it releases energy. And we'll be talking more about these as we go through other chapters.